about Australia, but once you leave Perth, the next city to Perth is 4,500 kilometres away. So we're in the desert, isolated, as far away from anything that you could possibly think of. Anyway, I'm really happy to be here, and we're used to the air travel, so that hasn't killed me. As Sandra said, Illumina acquired my company, Connexio Genomics, earlier this year. Connexio was started by myself and Damien Goodridge, who I think most of you would be familiar with here back in 2002. And really, we just started the company to supplement our income by providing sequence analysis software. We were both working at Royal Perth Hospital at the time, but it got bigger than our wildest dreams. And when we started, if someone would have said to us that your company would be acquired by the world's biggest biotech in 12 years' time, I would have told them that they were crazy. But what this signifies really is Illumina's commitment to transplant diagnostics. Illumina has become a massive company by selling largely into the research market, but they've made a commitment to change their business model a little and start providing diagnostics. Whilst this provides a massive business opportunity, I think it's a responsibility on their behalf that they do this, because they have the state-of-the-art molecular diagnostic instrumentation and scientists, and it's almost negligent of them not to become, or us to become, involved in molecular diagnostics. So this, I guess, is a message that Illumina is sending to the transplant diagnostics community, that we're in it, and we're in it for good. So why would Illumina acquire Connexio? And I think it's because we've got very aligned missions. Damien and I worked in the public hospitals, and we were at very much the interface of providing diagnostics to patients. And it was my job at the time to develop technology in order for us to provide the best HLA typing technology to our customers. And we were the only site in Western Australia that did that. So our mission has always been to develop solutions to identify the best donor, and I'll talk a little bit more about the best donor in a few minutes. But the idea really is to develop technology that would enable equivalent outcomes between haplotype identical siblings and unrelated donors. So to narrow that gap, everybody's seen the graphs that show survival and outcomes post-transplant comparing haploidentical siblings and unrelated donors, and there's a gap. And we wanted to develop technology that would allow us to narrow that gap. And it was this mission when we started the company that really drove innovation. Assign SBT sequence analysis software was our first product, but we'd also developed multiplex DRB1 sequencing-based typing technology, and putting these two together really launched the sequencing-based typing era back in the early 2000s. Also, the HARPS was our technology, and in recent years we developed the ability to do genotyping on non-coding sequences in Class 1. In the SBT era, you'll hear a lot of talks these days by people who are performing next-gen typing, describing how they've identified novel alleles in non-coding regions and how this is important. Well, we were the first to discuss the importance of non-coding polymorphisms in HLA Class 1 particularly, and we did that using Sanger technology. And of more recently, and a lot of what I'm going to talk today is about our product, GammaType. So everybody's seen pedigrees such as this, where we have two parents and two siblings, and both siblings have inherited the same haplotypes from their parents. So these are haplotype identical siblings, and they share the same identical MHC sequence over at least 4 megabases of DNA. There are recombination points at frequencies of around about 1 in 100 pairings, but essentially haplotype identical siblings share identical HLA sequence, and these are the best donors. An HLA match doesn't always mean a haplotype match. So in this instance, we have a patient and a donor that have identical HLA types, and they are, for all intents and purposes, HLA matched. However, the actual inheritance or the haplotypes may look like this. The patient might have haplotype 2540 or 103, and the donor have the haplotype 2540 13. So this is a haplotype mismatch. They're HLA matched, but they're haplotype mismatched. And this was shown to be important in a paper by Effie Petersdorf back in 2007, where they physically separated large segments of DNA and sequenced them to identify the haplotypes and perform haplotype matching between donors and patients. And they demonstrated in unrelated donor-recipient pairs, if they looked at those that were haplotype matched and haplotype mismatched, significant differences in the risk of severe acute graft versus host disease if you were haplotype mismatched compared to haplotype matched. So these are HLA identical, but haplotype mismatched and haplotype matched, or haplotype mismatched and haplotype matched. And as you can see, a considerable difference in the risk of acute graft versus host disease. So when I saw this paper, I was reminded of the work that I was involved with in the 1990s, 1900s. I'm not that old. In the 1990s of Professor Roger Dawkins' lab, who was my supervisor. He's actually not on this paper. He probably would have been one of the et al.'s, but most people here would know Frank Christiansen and Campbell Witt, and of course Dominic Chiron. But these guys back in 1995 showed that if you matched for MHC haplotype, you improved survival following unrelated bone marrow transplants. And they said in the paper that if HLA and MHC genomic blocks are matched, the survival is the same as identical SIBs. So that is the gap that we wanted to close. Therefore, what's important in unrelated bone marrow transplantation is not just HLA, but also the non-HLA MHC sequences. And this is what I want to try and talk about a lot today. HLA not so much as a functional protein that sits on the surface of a cell, but more as a marker for haplotypes and other sequences within the MHC which are contributors to outcome in unrelated bone marrow transplants. So that group was responsible for defining ancestral haplotypes. And what are ancestral haplotypes? So essentially, if you have two apparently unrelated individuals, myself and let's say Medhat. This is Medhat. If all you can see is the back of his head now, there you go. This is Medhat. Let's just assume that I have this HLA type, A3, B7, and DR15, and Medhat shares the same haplotype, and I don't have that, but I'm just using it as an example. And throughout the talk, I'll be talking about HLA largely as A3, B7, DR15, and that's old school nomenclature, but I'm using it so that I can fit it into a slide. So what is an ancestral haplotype? Two apparently unrelated individuals, and Medhat is originally from Egypt, and I'm from, although I live in Australia, I was born in Yorkshire in England. So if we backtrack through the generations, we'll come to a point where we both have a common but remote ancestor. So what does this mean? It means that if you share a haplotype with someone, you're related to them. Somewhere, back in time, through many generations, if you share a haplotype with someone, you have a common and remote ancestor. If you share a haplotype with someone, you're related to them. And they were words used by Marcelo Fernandez-Vina. So what is an ancestral haplotype? An ancestral haplotype is a haplotype which is conserved between unrelated individuals. The A1B8DR3 is a very common Northern European haplotype, also known as the 8.1 ancestral haplotype. This was sequenced in 19 unrelated individuals by Dan Garrity's group, Wade Smith in Dan Garrity's group. And they showed, even though the theory was that they were conserved between unrelated individuals, they sequenced 19 unrelated individuals and demonstrated that this was very much the case. So they only identified 3.8 SNPs per 2.6 megabases of DNA that they sequenced. So hardly any change at all. So according to the theory of Dawkins and the group that reported this stuff 10 years earlier, this was demonstrated to be true by this group. Now, some ancestral haplotypes are found globally, and some are restricted to regions. So the A1B8DR3 is found in Northern Europeans and is thought to be a recent haplotype, thought to have evolved somewhere around about 23,500 years ago. The A1B57DR7 is found in just about all populations and therefore comes from a remote ancestor which is much older than the A1B8DR3. The early work, very early work from Roger Dawkins' group, this is by Wenji Zhang of that group, showed that there were significant differences, physical differences between different conserved ancestral haplotypes. And this included copy number variants and SNPs and insertions and deletions. So, for example, the A1B57DR7 was the largest haplotype that they reported at the time, and A1B8DR3 was significantly different in size in terms in the region of tens of kilobases. So these haplotypes are significantly different between each other. And it was thought that the non-HLA differences would explain HLA disease association and increased risk of graft versus host disease in unrelated individuals. The mechanism is still not known, even though we know that A1B8DR3 is associated with many autoimmune diseases. No one really understands the mechanism behind it. But it's important to bear in mind that much of the MHC contains genes involved in inflammation and immune response. So let's look at this in a cartoon type of format, and if we look at ancestral haplotypes, the genomic blocks and recombinants in populations. So in any one population, if you were to look at the MHC within that population, a large amount of the population would have either a complete intact ancestral haplotype, or they would have recombinants at specific regions. So this individual's haplotype is a recombinant between the A1B8DR3 haplotype and the 37DR15 haplotype. So these recombinants can be quite complex, but then of course there's also many individuals within the population with an unrecognizable ancestral haplotype. And when this work was originally done in the 1990s in Western Australia, something like 80% of the population had a recognised ancestral haplotype, either something which is completely intact or a recombinant. I'll talk about these blocks in a minute, I think. No. So if we look at, from family data, family study data, looking at recombinants, it was very evident that recombination hotspots only occurred at specific regions, and they were somewhere between HLA-A and HLA-C and B, so recombination wasn't seen to exist from family studies between HLA-C and HLA-B. And then there was a region of the central MHC, which I'm going to focus on for the rest of the talk, and there was a recombination point between here and here, and then a recombination point between this gamma block and the delta block. So the Dawkins group named these the alpha block, beta, gamma, and delta, where HLA-A is a marker for the alpha block, HLA-C and B for beta, and so on. So when we're doing conventional HLA typing, we only type for, if we're talking about genomic blocks, we only type for the alpha block, the beta block, and the delta block. We don't have anything in this region. So that means that if we've got a patient and a donor, then the possible, and these are DNA sequence. So I've used yellow and black to indicate different DNA sequences. So this is a recombinant haplotype. So this could mean that if we see something like this, we could be seeing something where the sequence from this haplotype extends all the way to this block, and then they would be matched. Or alternatively, it could be that the donor has this haplotype all the way up until HLA-B, and the donor could have that haplotype sequence only up to the end of the delta block, which means that all of this region here has potential sequence mismatch. And whilst I'm using a cartoon to illustrate that, this gives you some kind of perspective of the amount of genomic information that could be mismatched between these individuals. So going back to here, this red line is indicated on this genomic map of the MHC. And you can see that the region for potential mismatch is significant, not only in absolute distance, but also the number of genes that exist in that region. So how can we improve haplotype matching in unrelated stem cell transplants, given that haplotype matching improves outcomes? What we really need then are genetic markers for all of the major MHC blocks. And if we think about HLA-A, the best way of obtaining the best genetic marker for the blocks that include HLA is to perform high-resolution HLA typing to the fourth field. Medhat and I had a discussion about this in the car, and I said, you can't get anything greater than high resolution. You can't have super duper high or absolutely brilliantly mega high. It's either high or it's not. But Medhat pointed out that really we're talking high resolution is defined by the NMDP as something completely different, and what we're really talking about is allele resolution typing. So what I'm talking about here when I say high resolution, Medhat, is that it's allele typing, which means that we are typing to non-coding level within the HLA genes. And we need to do this not only for HLA A, B, and C, but also for class 2. And we need to include as many class 2 genes as possible. But we also need, quite obviously, a DNA marker for the large chunk of DNA that exists between the beta and the delta blocks. So is conventional thinking about HLA typing sufficient to provide haplotype information in other blocks? I would say no, because you absolutely do need to obtain fourth field or non-coding resolution type HLA typing. And this is just 4 examples that we demonstrated at the last workshop, where we showed that if you did include non-coding polymorphisms, you're able to identify alleles which are specific to different haplotypes. So we identified a new 180101 allele that differed from the other alleles that I can't remember what non-coding region it was, but adding this 01 to 180101 was absolutely associated with the 35 DR3 haplotype, whereas the 18010102 was found exclusively on the 25 DR15 haplotype. Similarly for this allele, which was characterized by polymorphisms in non-coding, this was unique to the 44 DR7 DR11 haplotype, and so on. And we identified new alleles at non-coding regions, not only in HLA-B, but HLA-C also. And C04010105 is unique to an A11C4B35 haplotype that has DRB10103. This represented about 10% of the 04B35 haplotypes. What the absolute significance of that is, it's very difficult for us to know, but importantly, they are markers for completely different haplotypes which extend 4 megabases of DNA. So the polymorphism in a non-coding region that highlights the difference between 01 and O2 here is a marker for 4 megabases of DNA. So going back to the gamma block then, what could we use as a candidate molecular marker for that specific block? And we focused on the C4 genes. And the reason we did this was because the early work to characterize these ancestral haplotypes included complement allotyping. So these are genes that exist within that gamma block region of the MHC that sits between HLA-B and DRDQ. And as you can see here, we have different ancestral haplotypes, and the C4 genes are polymorphic. So these are allotypes as defined by electrophoretic mobility in an agarose gel. C4 is a protein that's not expressed on the surface of cells, I'm sure, as you all know. This is a very common set of assays in the 80s and 90s, but fell away a lot as the improvement in HLA typing started to develop. And also typing for these proteins is a much different technique to typing for HLA, the traditional HLA molecules. So just a little bit about C4. It's polymorphic, or the protein is polymorphic. Each individual, any individual can have between 1 and 3 copy number variants per haplotype, and each one of those copy number variants at the sequence level are greater than 99% similar. And these are tandemly arranged and about 10 kilobases apart. The genes are quite large compared to HLA, 41 exons, and are either 14 or 20 kilobases in length. So in order to develop a molecular technique for C4 typing, we had to get over the difficulty of designing an assay that enabled the separation of C4 genes, given that they were 99% identical. We kind of threw that idea out the window and said, well, let's not amplify them separately, let's just amplify all the genes together and we'll sequence them, and all we'll do is annotate the sequence differences. So if we sequenced homozygous cell lines, Sanger sequencing had to have the ability of identifying 1 in 3 nucleotides as indicated here, and this was pretty easy for us to do. So we did this on the haplotypes that I showed you in the previous slide, and we annotated the sequence differences against a reference sequence, and this essentially shows the sequence differences that we identified. We were really surprised to see that of the 24 different samples that we sequenced, there were 23 unique sequences, which indicated that C4 at the molecular level was highly polymorphic, if not quite so polymorphic at the protein level. So we then developed PCR SSPs for each of those polymorphisms, and then this became our gamma type assay. And the intent was that we would perform PCR SSP for each of those different polymorphisms shown on the previous slide and use them to determine match or mismatch between donors and patients. Just following up on how polymorphic the gamma type actually is, we looked at the SNP profiles in 96 individuals and identified 83 unique profiles. So if you take 96 different individuals and look at their HLA B and C, you're likely to identify far fewer different combinations than we identified for the gamma block. So this is highly polymorphic. It's highly interrogative of the gamma block, and potentially a really good marker for determining if patients are matched for the entire block using these as surrogates. So in our first study into determining whether or not this impacted outcomes in unrelated bone marrow transplants, we did a study with Noemi Farah in Curitiba. This is what we've called the Brazilian study, and we looked at 225 unrelated transplant pairs that were transplanted between 1996 and 2005. They'd been typed to the 4-digit level. 2/3 of them were matched 10 out of 10, 30, and a third of them matched 9 out of 10. So a third of them had an HLA mismatch. We performed the gamma type test that we developed on each of the individuals within each of the pairs. And we used a gamma type match as being a complete matched profile, and any mismatch represented a gamma type mismatch. So the results of that study start here. The first thing to note is that in HLA identical individuals, A, B, C, DRDQ, Luminex 4-digit, around about half of them are matched in the gamma block, and the other half are obviously mismatched. So that came as quite a surprise to us. We didn't expect so many differences between donors and patients. And I guess you would expect that if there's a HLA mismatch, which would be a surrogate for a haplotype mismatch, you would expect to see far fewer of these being gamma type matched. So then looking at outcomes, here we've plotted the probability of survival, severe acute graft versus host disease and chronic graft versus host disease for those that are 9 out of 10 matched and those that are 10 out of 10. And we were thrilled to see that the independent of HLA gamma type matching had a significant effect on the outcome of an unrelated bone marrow transplant. So we've extended these studies now and included a cohort from the Cleveland Clinic, and this was done in collaboration with MedHat when he was at Cleveland and before he moved to Baylor. We performed the same study, or used the same gamma block kits on 236 unrelated bone marrow transplant pairs. And each of these had been high resolution typed to ABC, DRDQ, DP, and also MIC-A. And the preliminary results of this were a little bit different. If we didn't identify the same types of outcomes if we just looked at single mismatches, but if we looked at in combination those SNPs that had an impact on mismatching had an impact on outcome collectively, there was a significant association with increased risk of severe graft versus host disease using a univariate analysis, with a hazard ratio of 2.43, which is something that's not even seen if you have an HLA mismatch. Importantly, there was the first publication of anything that of any of this type of work was performed by a South Korean lab who looked at a limited number of SNPs, and these SNPs were included in our panel, but they also showed outcomes similar to our gamma type mismatches. So these were looking at the Childers-Rogers polymorphisms and also the isotype-specific C4a, C4b polymorphisms. And if they identified differences in those polymorphic positions in C4, they also showed that if you were matched, you had a much higher probability of a better outcome than if you were mismatched in HLA identical pairs. So the next thing then was to extend this study, and we couldn't perform high-numbered studies using a PCR SSP because of the sheer volume and the speed in which we wanted to perform these studies. So we developed a next-gen PCR-based assay where this is the structure of the C4 gene, where we used a multiplex PCR assay to amplify exons 1 to 9, inclusive of the non-coding regions, then 10 to 25, 27 to 41. across PCRs 3 and 4. In some C4 genes, there's a 9 kilobase human endogenous retroviral insertion, and in other haplotypes, this isn't present, and we didn't include primers for this region. The amplicons are then sequenced and then analysed in a sign MPS, which later became the TruSight HLA product, which Sandra will talk about a little bit more in detail today, I think. So once again, we identified a sequence signature. So this is sequence positions within the gene, and then these are the sequence at those positions that are different from the reference. So here we've identified a C nucleotide, which is identical to the reference. Here a C and a T, and annotated that as a Y using the IUB codes and an M and so on. So we compared donors and patients' sequence profiles to determine whether they were matched or mismatched. What we didn't expect was the massive amount of polymorphism that we saw when we sequenced so many samples. So looking at the City of Hope data at 443 pairs that were HLA identical at high resolution, not HLA allele level, we identified 299 polymorphic positions. And in those over 800 samples that we sequenced, 132 of those 299 polymorphisms were identified only in one patient. So the dilemma for us then was, well, what is a mismatch? Is any polymorphism a mismatch? Or do we need to focus exclusively on those gamma-type polymorphisms we identified earlier? Or do we need to even include other polymorphic positions? In contrast to the HLA matched pairs that were gamma-type matched, we only identified 32% of the pairs had 0 sequence mismatches at all. So whenever we perform the gamma-type assay, which is a PCR SSP, in HLA identical individuals, we normally see around about 50-50. 50% are matched, 50% are not matched. But once we perform sequencing of the entire genetic region, excluding intron 9, of course, only 32% of those that are HLA matched are mismatched. And then there's a whole bunch of mismatched positions, all the way up to there being 27 sequence differences between 1 or 2 donors and patients. So in how we then take this into an assay that can be used routinely by labs, we really need to determine whether or not all mismatches are equal or not, because we don't want to exclude a transplant based on a single mismatch, which is a SNP that's evolved only in recent history. We are more interested in those SNPs that have evolved with the evolution of the haplotype, and it's up to us to identify which SNPs those are. So there's still lots of work to do with this, even though we've shown some clinical utility. Is any mismatch important? Are the number of mismatches important? Do we need to consider coding versus non-coding, those that are haplotype specific, and so on? The status of these studies, the Brazilian study has been extended to an additional over 100 pairs that have been typed on a MySeq using the next-gen assay that I demonstrated. They've been, and the original cohort has also been retyped to high resolution using sequencing-based typing. We just finished performing this study, and that data is being analysed. The Cleveland Clinic study is now under being analysed, as is the City of Hope. So in summary, I just want to show you this video. Can I get some help from one of the guys in there? I just want to
Where is the video? Can you look on the screen over there?
No, I want to access my folder down there. I hate showing my desktop. You know, it just shows you what a what a disorganized mind I've got. Sorry, I want to access. Oh, actually, I could do this. Okay, can we access that on there?
Project this one. ¿Cómo la proyecta ya?
Vamos a ver si ponen pantalla completa.
Está bien conectada, no es eso, es que tiene como dos pantallas.
Realmente con control F8 se puede duplicar, ¿no?
I've run over time. Maybe we won't do the video. I'll just do the summary and the conclusions. Can we go back to the?
You can project now the video.
Yeah.
This one.
We won't. We still have to do MedHat's talk, and I've got about 5 or 10 minutes to go. But okay, this summarises everything I've just said. Cue the sound. Don't worry about the sound. There's no talking, it's just crazy music. So just to summarise then, haplotype matching in unrelated hemopoietic stem cell transplants improves outcomes, including markers in the gamma block identifies haplotype mismatch pairs. The gamma block mismatching increases the risk of severe acute graft versus host disease. Complete resolution or allele resolution HLA typing and MIC-A typing, which I'm not too sure MedHat's going to talk about, can be used for identifying haplotype mismatching in other blocks, including the beta block. HLA typing as a genomic block haplotype marker requires including non-coding polymorphisms at the 4th field of resolution. And there is potential with next-gen sequencing to have a single assay that includes HLA, MIC-A, C4 and others. And you know, it should also be noted that the next-gen C4 sequencing data enables complete characterization of the C4 gene structure, which hasn't really been done before. And that would appear to be my last slide. If there's any questions, I'd be happy to take them. Thank you very much.
¿Alguna pregunta para David?
David, I have a question. When you define your non-coding regions, do you include UTRs and introns or just UTRs? How is that?
UTRs as much as possible, and all the introns.
So do you include all the introns and the UTRs?
Yep.
Thanks.
¿Alguna otra pregunta?
Otra pregunta.
Thank you very much, David.
You're welcome. Thank you.
Esperanza, ¿me ayudas a cambiar la computadora?
Oh, wow. Thank you.
Thank you so much.
Thank you.
Thank you so much for being here and share very interesting presentation.
Oh, my pleasure. Really, thank you.
Take this.
I just wish I could understand what
Yeah.
it said.
Hola, ¿tú ya mientras hago la introducción de Medat?
Perdón?
¿Podemos poner tu computadora mientras introduzco Medat?
Ah, sí, por supuesto.
We are going to set up the computer.
Okay.
Okay, so we are going to move forward with our program. The next presentation is clinical applications of HLA typing by next generation sequencing. And it is a great honor to have here with us today Dr. Medad Askar. Dr. Medad is the director of histocompatibility lab at Baylor University Medical Center.
This one? It is responding.
Morning. It is really a great privilege for me to be here and talk to you. And thanks, Sandra and the Illumina team for inviting me and giving me the opportunity. Like David, I have the limitation of not speaking Spanish, but probably if you guys invite me a couple more times, I become good at it. So today I'm going to talk to you about NGS, and we started actually playing with NGS when I was in Cleveland Clinic, probably around 2012, when I went to my department, my institute chair actually, I told him, there is this very cool technology. I think it will be very, very good thing for us if we get involved in. at the time, talking about NGS for HLA typing was like, talking about, doing testing on Mars or something. But he was very kind, and he actually indulged me. And I said, how much would that cost? They said, well, probably a couple of $100,000, and we'll need to hire a fellow in molecular biology and bioinformatics. So it's going to be probably $60,000-$70,000 a year. And he said, okay, yeah, let's do that. So, and actually, back then, there weren't really kits on the market or anything like. It was just a very abstract concept for people, to try things. I think we tried some of David's primers back then. You remember we did this on the Fluidigm system, and so and we did number of things. Most companies were just trying, and most of the reagents on the market, or they were not even reagent, they were just alpha testing, and they were not working very well. And actually, we were trying to develop our own, and then it turned out to be a bit major undertaking. It takes a lot of resources and a lot of commitment from big companies like Illumina to be able to pull this off. So we decided that instead of developing our own, we would be more involved with companies and just try their products. And in doing this, by the time I left Cleveland Clinic in 2015, we were just ready to submit our validation packet to the ASHI Accreditation Review Board to become accredited for NGS. And then I moved to Baylor, and part of my recruitment was to establish next-generation sequencing in Baylor. So I really am one of the probably few people who get to establish next-generation sequencing in 2 different labs. So I went through it, and it's really a very cool thing, because when you do it for the second time, you know exactly all the things you should have done the first time, but you didn't. So you get this benefit to do it. So actually, it took us probably 4 months just to implement it, and another 2 months to get accredited, to get ASHI accreditation in Baylor. And we were one of the first, I think, 10 labs in the United States that got accredited for next-generation sequencing. So I'm going to talk to you today a little bit about clinical implications of HLA typing by next-generation sequencing. And I will also, so I'll start by introduction to NGS as a technology. Probably you had a lot of this, but just to give you a very top view of NGS as a concept. And I will talk about the applications in both hematopoietic cell transplant or bone marrow transplant and solid organ transplantation. Because initially, even when we were trying to bring next-generation sequencing, everybody thought that this is high resolution typing, this is only for bone marrow, and there is no need to do this in solid organ. But more and more it became very obvious that there are a lot of advantages for using it in solid organs as well. And I'm going to highlight some of these applications. And then I'm going to talk about implementation and the laboratory accreditation if you were to provide this clinically. And I also had the benefit, in addition to implementing it in 2 different labs, is being involved with the ASHI Accreditation Review Board, who is the body that accredits labs to offer this test clinically in the United States. And I've been ASHI inspector for probably 15 years. I've been on the Accreditation Review Board for about 10 years, and currently I'm the division head of that department. So just a reminder, probably all of you know this very well, so that when we talk about the second field and fourth field and whatnot, so we are all on the same page. So this is the HLA, just the complex, if you may. This is the gene or the locus, and this is the separator to designate molecular typing. And this is the first field, or what historically we used to call the 2-digit typing. But now we are really moving away from number of digits, because it's more by fields, and originally each field contained 2 numbers. So when you say 4-digit typing, it automatically implied second field. typing, but now since we have second field for some of the alleles that are more than 2 digits, so now we are, to be more specific, you need to define which field level matching or resolution you are talking about. So this first field is the allele group, and in many times it corresponds to the serologic equivalent, although there are clear exceptions like DR3 and DQ3 and this sort of thing, and DQ7 and DQ8. There is the second field, which is the specific HLA protein encoded by this allele. And then after that, you go to, these are coding sequences, but they are synonymous. So these are substitutions at the nucleotide level that does not translate at the protein level. And then the fourth field would be the non-coding, the differences in the non-coding region. So when David said that these allele level typing or high resolution typing he mentioned are at fourth field level resolution, this means that they don't have any mismatches or differences in the non-coding regions or synonymous or the protein level or at any level. They are just exact sequences. And of course, this is the number of alleles we are talking about, and then as you can see, to just use the previous methods we were using, like Sanger sequences, it's going to become impossible. I mean, at this, when that was in June 2016, I believe, there was close to, there was above actually 15,000 alleles. And since then, there has been probably 100 alleles dumped into the database every week. So what do we do then? So earlier attempts, when we assumed that we will not be able to ever resolve all these ambiguities, was to say, okay, let's pick some of these alleles, and we're going to assume that these are the only ones that are common enough to assume they are present in patients or donors, and they share sequences, or they are different in sequences in the key, what we used to call key exon areas. So these are the areas that are assumed to have clinical relevance. Although that really, to my knowledge, and maybe David can correct me, this assumption has not been tested because we didn't have the data of those who are matched at the key exon. When they are mismatches, are there implications on on clinical outcomes? And really, it did reduce the thousands and thousands of alleles into 1,100. And this is the next generation, or the 2012 update. And you can imagine how many thousand alleles have been added since then. And it's about 14% of the entire database. And of that version 3.9, now we are on version 325.0. You may remember this when Bill Clinton announced the sequencing of the first human genome. And everyone was so proud that they did this. And anyone remember how much did this cost? $3 billion, yes. And how long did that take? Yeah, because you saw the slides. So, and actually, interestingly enough, when they made the big announcement in 2000, they actually didn't run any analysis. They didn't have it, so they just ran the test, the bench work, and it took them 3 years after that to complete the analysis. And why is that? Why it took them that long, and why it took that much? Because back then, high throughput sequencing was basically a form of genetic analyzer. That was the concept of high throughput sequencing. But now, high throughput sequencing is just you buy one instrument from Sandra and you're all set. You can run 5 full genome in one run, right? In 1 week, or probably less, and you get full genome sequencing at a cost of. So this is a publication that showed the cost per human genome sequencing, it started from 2001 to be, and that was using next generation sequencing, about 100,000. Is it this? 100 millions? Okay. And it went down all the way until in 2014, it was about 1,000. And this is really because of the throughput that next generation sequencing allowed. So this is publication, it's fairly old, it's considered old because in this field everything is moving very quickly. So I deleted I covered the platform names because now it is not true anymore. But just to give you an idea that the different platforms when were initially introduced, and this is at time of publication, and we are talking about hundreds, so this is 1,800 gigabase per run. So how that compares to the Sanger sequencing, you will be very surprised. Anyone can guess other than you, Dan, who saw the slides, where would be the Sanger sequencing here? Would it be here, here? Anyone have a guess? Where would it be? Any suggestions? Okay, so it would actually be here because that's all it can do. It's 1,800 on the average base per. So that's why this is the orders of magnitude difference in next generation sequencing versus Sanger sequencing. It's really very, very powerful technology. So how does this work? So this is just, if we spend a moment to highlight the differences between Sanger sequencing and next generation sequencing. So sequencing is by termination. It's basically when the PCR reaction goes on, it keeps adding nucleotide by this interaction that eliminates oxygen, and between the two these two nucleotide bases. But if we alter them by adding, removing the oxygen molecule from one of the nucleotide in this position, then when this gets incorporated, then the following nucleotide cannot be incorporated anymore, and that terminates the reaction. So basically, if this is happening and we include mix of normal nucleotides and these altered nucleotides, they will get incorporated at different positions at random. And then you would end up with chains that are terminated just by random at every sequencing position. So you would know which position has which nucleotide, because we're gonna label each of the 4 nucleotides by a different dye. So basically, we would know that all of these positions have a C in them. And then if we compile all chains that terminated with all dyes, then we would know what the sequence would look like. And we run, so these sequences gonna be different by just one nucleotide. So the molecular weight difference is going to be very small, and we cannot run them on just regular gel. It wouldn't detect the differences. So we would need capillary electrophoresis to be able to separate these sequences just based on one nucleotide difference. And that would end up with a resolution looks like this. And if anyone thinks that this is not high resolution enough, you may want to look at other sequencing kits that was available at the time. You will see that this is really very decent resolution, but still it is not high resolution enough. And we needed to run a bunch of other tests to rule out all of these common and well-documented alleles that are required by National Marrow Donor Program, or NMDP. So you have those, we had to run SSO, and we had to just pretend that those do not exist because they are not on the common and well-documented allele. Although now when we do next-generation sequencing and we get fourth field resolution, we encounter many alleles that are not on the common and well-documented allele. And just ruling them based on this assumption historically was not appropriate. And we have a bunch of other ones that we also rolled by SSO. So that would be, the typing would be any of these pairs. And this is really because we could not have phase typing. So we looked at different exons, but we could not phase them. So if we know that this possibility, it's either this one and this one, or A2403 and 3105, and we look at the SNPs, each one of them alone wouldn't tell us. But if we phase the entire sequencing, which we can do now, then we can tell unequivocally whether it is this possibility or this possibility. Am I going in the wrong direction? I'm sorry. Okay. So that ends actually with this very unambiguous typing that oftentimes, in fairness, is going to be the common and well documented, but not all the time. So in this particular case, that was actually the commonest haplotype, and we have thousands of reads that support this typing. So just the workflow in principle. So we start with initial sample amplification where we extract the DNA. We use the primers that is going to specifically amplify the HLA loci of interest, and this is through the target enrichment. And in general, there are two approaches for target enrichment. Basically, have a DNA that is mainly of the, or only of the loci we would like to test by next generation sequencing. And one of them is to have this whole gene approach, and you start from the beginning, or it depends on how far out you can go, so that you include all exons in one stretch. Of course, this is going to generate this giant fragment of DNA that is not appropriate for sequencing. Then you start to use enzymatic fragmentation or other methods so that you can separate it into small fragments that are suitable for sequencing. Or alternatively, you can use exon-based typing, targeted exon, where you have multiple amplification reactions. Each one of them would amplify part of the gene. The downside of this is that you lose phasing. So you end up with the same problem we would have with Sanger, that you cannot tie which polymorphism goes with which one, and may lead to possible higher level of ambiguities. And then once we come up with these fragments, we need to do library preparation or library construction, where we generate fragments that are labeled with specific barcodes that allow multiplexing, so that we can add multiple samples from multiple patients, allow pooling of samples together from multiple patients to generate this high level of throughput. And then once we get to this, we do the sequencing by loading them to the sequencer. And then we do the analysis. So the fragmentation, as I said, simply these giant fragments, we want to fragment them using enzymes into small pieces amenable for sequencing. And then we add what is called overhangs. So basically it does two things. We need to add barcoding so that we allow pooling of samples from different individuals to be tested together. And we need to do adapter ligation so that these fragments can be specifically, can bind to the solid phase of the next generation sequencing platform. In the Illumina platform, it is the flow cell. So basically, we add these barcodes. So in one sample, it is going to be this blue. In the other sample is going to be this yellow. So basically, it is going to be short sequences unique to these barcodes, so that when we mix all samples together, we know which fragment came from which sample. And the other thing is to add these adapters that is going to bind to the complementary sequences on the solid phase. And then after that, some of these still, since they are fragmented enzymatically at random, some of them still may be too short that wouldn't give you enough information. I would just crowd the flow cell unnecessarily and use the real state of the flow cell. And some may be prohibitively long that are not gonna bind very well. So we do size selection. So we optimize the process so it sequences the fragments that are most amenable for sequencing and giving high quality information. And there are multiple ways of doing this, and this talk is not unique to this product. So these are some of the things that may be used in this protocol, may be used by other vendor protocols like the Blue Pippin, which uses principles of electrophoresis. But you can also do this without the particular instrumentation using the impure beads that select a specific fragment size. And after that, not necessarily after that, throughout the protocol, you need quantification. So you need to make sure you don't want to go all the way through the process and end up with no product. Then you wasted time and you wasted reagents. So in an ideal world, you would like to make sure that every step of the protocol still you have enough DNA quantity and the quality to proceed to the next step. But if you do that, the protocol becomes 10-day protocol, and you don't want to do that either. And in another ideal world, your protocol is so robust that you know that every step is just going to generate adequate DNA quantity and quality, and you wouldn't need to do any qualification. A happy medium in between is to have as little quantification steps that allow you really to have to have enough product of adequate quality and the quantity. And I think the current protocol has only one step of quantification. Is that correct? Okay. So basically, just historically, what we used to do in the Sanger days, if we wanted to do a new allele, it was just to do hemizygous sequencing, just one allele, which was a requirement to report any new allele to the IMGT database. We would just do cloning through bacteria that some of the bacteria are going to pick up both alleles, some going to pick allele 1, some going to pick allele 2 at random, and then we sequence those that have just one allele. And that would tell us when we see these ambiguities, it could be this or that. It becomes a question of the C and G, which one is in allele 1 and which one in allele 2. But if you do the cloning, then you know the hemizygous sequencing. But now, with next-generation sequencing, you could actually achieve something very similar by using this after you do the fragmentation and doing the barcoding and the adapter ligation. You could see exactly which fragment came from where, and then you do this clonal amplification that allows you to generate hundreds and even thousands of copies of that very specific fragment. And then while these are formed, so you do this by immobilizing them to the solid phase in the Illumina technology, the flow cell. And when every nucleotide is being added, a video picture is being taken, and that gives you which nucleotide is being added. And this shows you the amplification process. It starts with each fragment with this adapter is going to bind to the complementary sequence on the flow cell, and then the PCR reaction, and it makes this bridge amplification, and then you do the naturation, so you end up with that many copies of the same fragment, and then the process repeats 100 to 200 million times. So, and this, every time you add a nucleotide, you are taking a picture, and you keep adding another one, and you take another picture, and you compile all pictures together, and that generates your sequences. So when it comes to analysis, you would hear different vendors talking about, multiple algorithms and whatnot, and some say you have 5 algorithms, and they have 10 algorithms. But actually, in a very simplistic way, at least in my mind, an algorithm is what generates independent genotype call. So in this algorithm, you may use multiple sub-algorithms, if you may, but categorically there are known 2 analysis algorithms. One of them is de novo assembly, which basically it doesn't rely on reference sequence. It's just it's like the Humpty Dumpty type of thing. And of course, I'm very shy to talk about this when we have a programming guru here. He says, this guy just doesn't get it. No, that's not what we do. But you throw the sequences, and based on sequence alignment. So this is not complementarity, although these lines usually imply complementarity, but we're really talking about sequence similarity or identity, right? That's right, David? And then based on this, you create this sequence and genotype. In the reference-based analysis method, you basically start with a reference sequence that you bring from the IMGT database, and you see the best fit for each fragment. Where would it fit on this reference sequence? So NGS key features, one is massive parallel sequencing, and that really allows expanded gene coverage, because historically, as we said, we used to do only key exons, not because we thought it's a very good idea, it's simply because that was, with the throughput allowed by Sanger sequencing, that was the only practical thing to do. Otherwise, we would have taken for typing each patient years and years, like the Human Genome Project took, and it would have cost thousands and thousands of dollars. And the high throughput, so the massive parallel sequence allows high throughput. And the other thing is clonal amplification, which allowed phase polymorphism information. And that's using the ship. And these ships, it's amazingly small. It's really very impressive how these technologies work, doing all of these thousands of amplification reaction on such a small physical structure. And if you have expanded gene coverage, you have phased clonal amplification, that leads to less ambiguities and ideally gives you high resolution or allele level typing. And the high throughput allows low cost per typing. And that's really was our argument for the administration to implement next generation sequencing, not only for bone marrow, but also for solid organ. Because we showed that if we do next generation sequencing for everyone, bone marrow and solid organs, donors and recipient, the cost decreases dramatically and even becomes lower than the Luminex and the other comparable technologies. So as you can tell how powerful this technology is, for the first time in Nature Methods, they named a method as Method of the Year. So that was the first time to call any method Method of the Year, and that was next generation sequencing. So now, apps. Let's talk about the applications for this technology. Is this technology relevant to transplantation or any clinical field? So this is a study that we did in the Immunobiology Working Committee when we looked at a cohort of bone marrow transplant recipients and their donors, and we correlated this with outcomes. And it showed that allele level typing unequivocally associated with all major transplant outcomes. It is associated with lower severe graft versus host disease, lower transplant-related mortality, and better overall survival. And this is a study about HLA mismatches associated with unrelated bone marrow with reduced intensity conditioning. So the previous study was mainly with fully myeloablative, and the similar results were shown with reduced intensity. And the message here is really, regardless of what treatment modality in bone marrow transplantation, allele level typing is not going to be something you can overcome. Next generation sequencing is not only applicable to HLA, it's a technology applicable to all genetic loci. And KIR is one of the loci that have been shown in a number of high impact publications to be associated with clinical outcomes, particularly in the area of leukemia relapse. So this is one of the New England Journal of Medicine articles showing that the prevention of relapse associated with having activating KIR2DS1 receptor. And this is another study in the Journal of Immunology that showed that HLA and KIR genotype can predict low affinity interactions that are associated with lower relapse of hematopoietic cell transplantation for acute myeloid leukemia. So I said that historically with Sanger sequencing, we used to be able to sequence only for exons, what we called the key exons, which were exon 2 and 3 for class 1. And these are the areas that encode the peptide binding groove. And it is thought that this is the part that is most immunogenic, most exposed to the immune system, and the part responsible for graft versus host disease and responsible for graft versus leukemia effect. So there was this convenience agreement among people in the HLA typing and clinician that that's what we're going to do because this is the most feasible thing and we're going to move on with our lives. But now with next generation sequencing, we really don't have to do this. We can sequence the whole thing. And now for the first time, we will have data enough to go back and see those who were matched at key exons, but have differences or mismatches at other parts of the gene. Did they behave differently clinically? Historically, we would have not been able to answer this question, but now, since more and more patients and donors are typed by next-generation sequencing, we will have the data to answer this question. So, parts of the argument for non-coding sequence, there has a number of publications that showed that some of the sequences in the untranslated regions, 3' and 5', really have been associated unequivocally with different levels of expression. So how this is relevant? So this is a study from the HIV literature that shows that differential microRNA regulation of HLA-C, so now we are talking about C locus, so it is known that C locus in general have lower expression. But what wasn't very clear until recently, that it is not only have lower level of expression, the level of expression is more highly regulated. So it can ranges from very low level of expression to very high level of expression. And did that make a difference? And that actually was encoded by sequences that are complementary to microRNA that in the 3' untranslated region. So we would have never seen that if we were only doing exon 2 and 3 as the key exon. So, and those were associated, the people who have the higher expression, these had higher inhibition of the KIR, and those had lower ability to fight HIV. So it all makes sense. Would that make a difference in clinical outcomes in bone marrow transplantation? So this is just to show you the difference in level of expression based on these sequences in the untranslated region. And here you see this is the fold change of relative luminescence, and it goes from 80 to 200, so almost 3-fold level of expression. And that level of expression was associated on level of HIV control. And another, so when we tested this in the context of hematopoietic bone marrow transplantation, it was clearly that HLA-C expression levels define permissive mismatches in bone marrow transplantation. So if you have to mismatch for a C antigen or C allele, it was shown clearly that if you mismatch for a low expression level, then you have better outcomes than if you mismatch for a high expression level. I think that is not hard to believe. It makes perfect sense biologically. And since now we have this ability to sequence the whole gene, we can get the information about the different levels of expression. And most recently, there was this publication from the New England Journal of Medicine where they showed that level of expression in DP, which also another locus that is known to have constitutive lower expression compared to other class 2 genes, that it seemed to also be highly regulated, and there are clear differences in low expression versus high expression in terms of impact on graft versus host disease. And this is really fascinating to me because, so let's look at this one. So if this is your donor and this is your patient and they are mismatched for this is the DP mismatched transplants, okay? So you have mismatches. You have here the green, and then instead of the blue, you have the red. Or here you have the red and the blue. But if they are both, the mismatch are low expression, then you have low risk of graft versus host disease, okay? That makes perfect sense. And if we look here at if the mismatch happen to have higher level of expression, then you have significantly increased risk of graft versus host disease. Not only because the DP itself is different, but also because here you have, since this is high expression compared to this one, you have higher opportunities of presenting peptides and differences in the minor histocompatibility antigens. So it is not surprising when in the mismatch, when you have higher expression versus lower expression. So if I have to mismatch for DP, I will have higher risk if I mismatch for high level of expression. And we get this information from next generation sequencing. But look at this. This is really the fascinating part. In HLA-DP matched transplants, okay? So this is matched. And here, if you have lower expression, you have low risk of graft versus host disease. But look what happens when we have high level of expression. You have higher risk of graft versus host disease even in HLA-DP matched. And why is that? Because now you have higher chances of presenting to the immune system different peptides and with more differences in minor histocompatibility complex. So really these non-coding sequences may turn out to be even more clinically relevant than what we used to match for, which is the antigen itself. So this is really remarkable. I think our whole way of thinking around matching and around compatibility between donors and recipients, when we decipher these non-coding differences, may really change completely, and we may stop matching for what we used to match. Of course, that's one extreme; it may never happen, and start matching for gamma type and all things that we never even knew they exist before. So it is really fascinating time for us to be investigating these things. And this is the publication that David showed you about haplotype matching. And I'm not going to belabor the issue because David explained it very well, the differences. If you have multiple otherwise genotypically identical donors, is there any difference of pulling one versus the other? And it was shown that if you pick the haplotype match, you get better outcomes, and especially in the severe graft versus host disease. So there is also, this technology is very powerful that it is not only applicable to DNA, it's equally applicable to RNA. So in this way, we can actually sequence the T cell receptors. And the fascinating part about this, you may remember that with all protocols for hematopoietic cell transplantation, bone marrow transplantation, the protocols that eliminates a lot of immune competent cells to like the depleting protocols, like T-cell depletion protocols, they lower the GVHD, but they increase the relapse rate because they also lower the graft versus leukemia effect. And that was the dilemma, is that the same cell that causes graft versus host disease is the same cell, it is the T effector cell, that causes graft versus leukemia effect. But actually, this is not, and we kind of settled that this is how life is, and we have to find the balance between depletion and non-depletion and whatnot. And we would do crazy things. We do depletion and we do add back. We add the cells back, all kinds of things. But actually, it doesn't have to be this way, because it is true, it is the same cell type, but it is not the same cell clone. So there are cells that have T cells, that have T cell receptors that are specific to the tumor antigens and would cause graft versus leukemia effect, versus other clones that are reacting against normal native cells and would cause graft versus leukemia, graft versus host disease. And by sequencing the T cell receptor, we can identify which clones are specific to graft versus host disease, and those can be eliminated without impairing those who affect graft versus leukemia effect. I don't think this came to materialization yet, but I think this is really the direction where we will be going, especially with next generation sequencing. So now, I hope I convinced you that there is a lot of current and future applications for NGS in bone marrow transplantation, but then why bother in solid organs? Solid organ, we don't even match, okay? We just, because of the shortage of organs, we would like if I have a heart patient who is having advanced heart failure, who's gonna die today, and there is a donor that is mismatched across all antigens and has a healthy heart and everything, I'm not gonna leave this heart and let the patient die waiting for a perfect match. So, but how this becomes relevant in the context of solid organ transplantation? One very interesting study, it was calcineurin inhibitor elimination protocol. Basically, the dilemma with solid organ transplantation is many of the drugs we use have very high side effect cost. Some of the drugs we use after kidney transplantation are nephrotoxic. So we put a new kidney in and we give the patient daily drugs that would kill this kidney 100 nephrons at a day, or number of nephrons at a day. So there is always this notion that we want to eliminate the use of these drugs. But with many of these trials to eliminate these drugs, when we do that, these patients reject their kidneys. So we say, you know what, let's kill it one day at a time rather than just let it die right away. Neither is ideal. So there was this clinical trial, and they did it, and they looked at patients who were low risk, low immunologic risk. First time transplant, no antibodies. They didn't develop antibodies on calcineurin inhibition for the first 6 months. Everything looks perfect. So they said, okay, let's do this. And these are the controls. So just to look at the diligence. So these are complete withdrawal, the green axis. And these are development of donor-specific antibodies, the blue arrows. And these the blue triangles, and the red triangles are the development of cell-mediated rejection. So they wanted patients, so these are the control arm who remained on calcineurin inhibitor, and these were the patients who got withdrawal. And the goal was to to have patients start on withdrawal if they didn't develop any donor-specific antibodies or cell-mediated rejection. And here we can see it really doesn't take appreciative, nephrologists to say, or immunologists to say, that many of these patients developed DSA and developed cell-mediated rejection. So actually the NIH stopped the clinical trial, said, no, you can continue to do that. And in retrospect, when they tried to look, there were some of them that really did not develop DSA or developed cell-mediated rejection. And the differences were, so this is the breakdown of the study, so I'm not going to bother you with these details, but let's look at this slide. So here, if we look at these patients, so all of those are the patients who got withdrawn. And these are the patients who did not develop DSA, and these are the patients who developed DSA. And here you can see the number of molecular mismatches, if you may. These are motifs, they call them iplets. These are protein motifs in the HLA molecule, and they compared them. And you can see that in the ones who didn't develop DSA had very low molecular mismatching level. And when I asked Peter Neckerson, who gave me these slides, how did you do allele level typing on these patients? Because that's not the standard in solid organ transplant. He said, I ran SSP. Told him, how long did this take you? I mean, you would need to build the monuments of thermal cyclers to do hundreds of patients by SSP. And he said, yeah, it took them a couple of months. And I told him, you should have sent them to me. I would have tested your 500 patient on just one run. And they give you allele level typing by next generation sequencing. And we joked about it. So now I told him, if we were to repeat this clinical trial and we want to see which patients to randomize to withdrawal, I wouldn't randomize, I would include your initial criteria, but I would put a criteria that patients who have high level of molecular mismatches based on allele level typing, ideally by next generation sequencing, those who have high level of mismatch, I would not attempt withdrawal with them because those gonna be destined to have or have very high risk of developing donor-specific antibodies and rejection. And he agrees that this is probably a good thing to do. So I would like to spend some time on implementation. So now I hope I convinced you that next generation sequencing is a very good thing to do. So how do we do it? Okay. So these are the things that you need to think about. The reagents, the laboratory workflow, the bioinformatics workflow, the staffing, the space or construction, the vendor support. And if we talk about kit configuration, I'm not going to cover everything. And also, I'm not implying that anything I say is unique to any particular kit. So there may be some of the features shared by other vendors. And really, it's up to you to see the collective constellation of considerations that gives you the highest advantage, which kit would be representative of that constellation. So the chemistry. So one of the things we need to look at is the read length. Because if your goal is to give phase typing, and you end up with these short fragments, they may not be able to align well, and they may not be able, and you may lose phasing in your typing. And pair end reads, which really, this is something very powerful in the Illumina technology, is that it allows you to sequence each fragment from both sides and gives you two readings simultaneously. And the error rate. And each technology platform has known error rate, and this is not necessarily sequencing genotype assignment error rate. It's just incorporating the wrong nucleotide just by sequencing error. So this is just to clarify the concept of pair end read. If you have a fragment that is 400 base pairs and you are using read length of 200 bases, then you are ensured to have overlap and coverage. If you increase it, the more you increase the fragment length, that could lead to loss of phase. However, there are bioinformatic approaches that can make up for this, but it's just that this is something you need to be mindful of. Also, there are publications that shows the rate of the error rate, percent error rate per number of nucleotides incorporated, and plots this against the read length, because the longer the fragment gets, the higher, at least in many technologies, the error rate becomes. And in this publication, it was shown the red here actually is the Illumina-based platform, and this is another platform. And it shows that really it has the lowest substitution rate, the lowest error rate, the lowest insertion error rate, the lowest deletion error rate, and the lowest, consequently, the lowest overall error rate. So this is one of the things you really need to consider, which technology to apply. In an ideal world, you would like technology with the lowest error rate. The other thing to consider is what we talked about in terms of whole gene versus exome-based. So exome-based usually is very good at giving you two field digits, but if you're looking at intronic sequences, non-coding sequences, then this is not probably going to be this. And really, it's a place where you sit and think what you are doing typing for. Is this initial screening for donor registry? Then you're going to do after that a specific more extensive typing, or you want to get the full results from the beginning, because there is a cost for doing everything. And this is just to show you the different genes and different coverage and things of that nature. So if we talk a little bit about laboratory workflow, these are things you need to consider when you are evaluating different protocols. What is the total time? So when they say that this is a 3-day protocol, or this is a 2-day protocol, or this is a 5-day protocol, of these times, how many hours are hands-on tech time? Because it makes a difference if it is a 3-day protocol with, I'm making this up just as example, with 8 hours hands-on versus 2 days with 12 hours hands-on. Is it more expensive? Is it, and it really becomes, are you in a rush? Your clinical application to give you very tight turnaround time. So really, these are all things you need to consider when you are evaluating different kits. And as I said, robustness of the reaction. And robustness of the reaction is going to reflect on two things from practical standpoint as someone who is in the lab. The DNA requirement. Different kits on the market require different DNA concentration to start with. And in an ideal world, you would like robust enough protocol that would allow you to use as less DNA as possible, so that when you come across these patients who are bone marrow transplant patients who had, chemotherapy and whatnot, and they do not have white cells, and you have very little DNA, then your protocol can work for them. And the other one, as I mentioned, is how many quantification steps. And also the forgiveness of requirement. So there are protocols that are really very stringent in the requirements. So when they say you start with this X amount of DNA at the beginning, if you use less than that, that reaction just doesn't work. Or the fragment length, you need to have a very tight range of fragment length. But there are protocols that are robust enough that they are forgiving. So it allows you to use larger size range of the fragments, which really allows you to get better phasing information. So all of these things you need to look at when you are considering which platform to implement. Instrumentation and the automation. So this really comes to what pieces of equipment you will need. And you really need to give it a serious thought about manual versus automation. And it depends on the throughput, the volume, and the cost of implementation and the cost of labor. And this might differ from one country to another. But these all things that just need to be considered together. And what I call, so when I presented this to the financial leadership in Cleveland Clinic and after that to Baylor, I modeled typing. So I provided to them different models. If we use labor, manual labor with no automation, and based on this throughput volume, that would be the cost per typing. If we use automation, that would be the cost per type. If we use part automation, part manual, that would be. If the volume doubles, then that would be the cost. And really, you come up with a model that really everyone thinks that this is really what makes most sense based on the institute circumstances. And of course, the capital cost. And one thing you may consider is, although it's becoming less of a concern, most automation companies now have already written scripts. It could be involved a little bit, but that used to be historically the case. The bioinformatics workflow, this is really some of the reagents on the market require analysis from servers and a specific IT infrastructure that comes with cost, initial and maintenance cost. How you can handle protected patient information issues, especially in the United States. So this is really a big contingency. The storage capacity, so these runs generate massive amounts of data. So, and there is accreditation requirement to have all these stored for at least 2 years. And not only, and you need to store all raw data, the FASTQ files that allow reanalysis. You may want to consider, if you have enough storage, to store also the analysis files so that if you needed to, if you lost the data, you don't have to run the analysis again. Network interfaces, data transfer, all these things needs to be considered. The specification of every piece of equipment, it needs to be compatible with the requirements from the vendors and the analysis software. So for software, it's important whether it runs locally or on a server. And in ideal world, it would be flexible enough to do both. Multiple algorithm versus a single algorithm. I would say that although initially the thought was there was a need for having multiple algorithms, and actually I personally was on the committee that wrote the initial version of the standards for NGS, and we had this as a standard for ASHI standards, and then we changed our minds and we actually made it just a recommendation rather than a requirement. And I would say if it is one algorithm but it gives you the same level of concordance of the genotype assignment, then what do you care? How did we come up with this assignment? It's just one thing to think about, and when you hear people talk about it, just take a step back and see why we are doing things the way we are. I think at the time when we did this, was it 2014, something in this time? I don't think the reagents, the software in general were robust enough at the level they are today. So probably that was a need back then. It may not be at the time. Also the analysis time. There are differences. Some of the softwares takes, couple of hours, some of them take 6 hours, and that all is going to affect your turnaround time. And automated upload and starting the analysis. So you want, if you are leaving your MySeq running overnight and it finishes, at 4 a.m. or at, midnight, you want the data to be captured automatically to the software so that when you come next morning, you don't wait another 2 hours or 4 hours or whatever that may be. You already have your analysis done. And you need user-friendly layout. So the amount of data you get and the quality metrics and it could be overwhelming. So it's always a balance between having in the same view, because you also don't want to keep clicking from one view to another, especially this is high throughput technology. If you are reviewing a run of 96, which is what I do, it's really, if you're going to spend 3 minutes on each sample, it becomes, like a full day time job. This is really very critical to come up with this, that the balance that gives you all the information as little number of screens as you can. In an ideal world, you would like intuitive flagging system that, again, not overwhelming, but this does not run the risk of you missing some of the quality metrics not being met. So quality metrics is a requirement by ASHI standards and by all next generation sequencing standards. You need to identify what are the quality metrics that affect the integrity of the run. At what point you say this sample needs to be repeated, and at what time you are confident that yes, this is the genotype, and if we run it 10 times, there is no point of repeating. This is what this. And at that point, it's either that you can confidently release the result or confirm by another method, which is also required by the standard to have a backup method. Although who knows, with the development of the technology, it may be the case that at some point we won't need the requirement for a backup technology. Homozygosity, and this is really very critical because there is this phenomena of allelic imbalance. Not both alleles at the same locus gonna amplify at the same efficiency. So you may end up with 50, in an ideal world, you would like to see 50% reads from allele 1 and 50% reads from allele 2. And that happens quite often. But sometimes you see 60-40. Yeah, that should be fine, right? Sometimes you see 70-30. Yeah, that's okay, right? How about 80-20? Okay, yeah, we can live with that. 90-10? 95-5? Where we draw the line? At what point we say, no, there is no second allele. This is just sequencing noise. Okay, so really it's critical that in your validation set to include as many homozygotes as possible so that you see what the strength and limitation of your software and at what point you are confident that this is the right call. Novel alleles, and this is really important to be able to see how this is actually, I think, a requirement by the standard to have flags for novel alleles. Discordance between the two algorithms, if you have multiple algorithms. In an ideal world, you would like to see a distinction between common and well-documented, because for clinical purposes, that's a requirement to rule out, regardless of the technology, the NMDB doesn't care. You still need to rule out alleles that are common and well-documented. So in an ideal world, the software would highlight, either make them bold or... So you know that you have common and well-documented that is not ruled out. And also one very cool thing is if you could have flags for uncommon haplotype association. So you see that allele and DR and DQ that normally do not come together, then it just makes you, not necessarily that it cannot happen, we see this quite often, not quite often, but it is not uncommon thing, then you would take a closer look at the data. Is there any change I can make that would allow this to become more haplotype common association? And the goal of having as many quality metrics and indicators is really to reach a level. This is a high throughput technology. It's possible that you may be running, or at least in some of the registry NMDB labs, that would be running multiple 96 samples run a day. So in an ideal world, you would like to see a samples that met all quality metrics. No uncommon and well-documented. Everything is common and well-documented. All associations are imperfect haplotype association. Then probably it's okay not for anyone to look at this and just auto-sign it out. So that's in an ideal world. I know this is extremist view. Some people might not like it, but I think we did this actually. We did some study and we looked for what is the percentage of wrong misassignment in samples that met all quality metrics and were no common and well-documented allele and no uncommon haplotype association, and it was 100% concordant without anyone looking at it. So when we went back and we looked at it, it turned out to be 100%. So staffing, as I said, modeling your workflow. How are you going to do this? Number of runs per week. So these are considerations. And this is going to be determined by the overall throughput, but also by turnaround time. So it's possible that you patch everything in 96, but then if your lab receives only 20 samples a week, you're not going to be able to run it only once a month. You will need to have smaller runs. And that will determine the number of samples per run. Total hands-on bench work time, analysis time. So what would be the appropriate staffing and what would be the time to competency? And really that again comes to how involved the protocol is, how many steps, how many hands-on time. That is going to determine what time to competency is going to be. The more straightforward the protocol, the less time to competency would be. And in an ideal world, you really need people with good molecular skills. So, and it's even better if you have HLA experience, even better and better if you have SPT experience, and NGS experience is always good. And the best of all worlds, if you have people with NGS HLA experience. But, if you find a bunch of them, please let me know. I would like to hire them. And space and construction, you really need to think this through, because you're going to add instrumentation, and each instrument is going to have its own footprint and its own requirements for setup, and you really need to account for all of that. You're going to need computer workstation for the analysis, because even if you're going to do auto sign out, you still need someone to determine which samples are eligible for auto sign out. And you're going to add local servers. Are they going to be housed in your lab, or are they going to be in IT space, or how this is going to work? Your clean room becomes an issue when, if you want to introduce automation in the preamp area, many of the I inspected more than 50 labs around the world, and many clean rooms I see would not fit a robot. So this is something I'm going to get a newer clean room, or how this is going to work, because then that becomes a critical issue for automation. Vendor support. So really, I found this to be very critical thing. You're gonna need the vendor because this is very fast pace evolution technology. All protocols are being improved, and you would need that. So it is going to be a long-lasting relationship, if you may, with the vendor, and you don't want to be in the wrong relationship. So initial training, this is critical. How much support are they going to provide? How much are they gonna get you to a level where your techs really can go on their own? Because showing it to them, especially if your techs have not been involved in next generation sequencing, showing it to them how it works is going to be helpful, but not that helpful. They really need to make sure that they can run it on their own. Access to troubleshooting. So you would like a company that when you have a problem, you just email someone, call someone right there and then, and to hopefully get a response no later than next business day. Because otherwise, you have a run in the making, and it's just no one getting back to you, or they can't figure it out themselves, or whatever the case may be, then that really becomes problematic. Access to multiple levels of the company, because you're going to have all kinds of problems. Even if you have been doing this for years and years and years, you're still going to have all kinds of problems. Not problems that the technology is problematic, but here and there, there are going to be things that you haven't seen before. And you don't want to make the wrong assumptions. You need someone who is very knowledgeable with the kit and knows its strength and limitations, so that instead of you going by trial and error, even if this happens once a month, you don't want to do that. You want someone who already figured it out and tell you what to do. So you need access to molecular biologists. If it is something bench work related, we didn't add this, how we can salvage this run, can we add it now? This sort of thing. Bioinformatics, you get the analysis, something the software doesn't make sense, you need to have access to these folks as well. Sales rep, it's really, you need, you run out of reagents, you wanted them earlier, you wanted to delay them because you are running slower than what you expected. You really need to have this communication with sales rep. So validation quickly. How are we doing on time? Are we late? Yeah. So I think we probably stop now, and if there are any questions, I can answer. So thank you very much.
Thank you very much for that. ¿Alguna pregunta? ¿Algún comentario? And Dr. Medad is going to be with us tonight during our dinner, so you can still come with some questions, some comments, and take the advantage of this opportunity.
Thank you.
Thank you so much.
Thank you.
Yeah. And in case you want to see you have the slides also printed in your manual, so please don't hesitate to send me any
absolutely.
question by email.
And I think
or
I have my
whatever, and I
email,
can-
so if there's anyone after that has any questions about validation or implementation or accreditation, please feel free to email me or email Sandra, and she knows how to get a hold of me. I'll be glad to answer any questions.
Thank you so much.
Thank you.
This is for your participation. Thank you so much.
Thank you.
Bueno, nuestra siguiente presentación, vamos a tener un cambio en la agenda. La siguiente presentación es a cargo del doctor Humberto Valenzuela, que nos va a hablar acerca de aplicaciones de HLA en infección por HIV. Sin embargo, vamos a hacer un cambio. Tenemos a las 11 también la inauguración del laboratorio de diagnóstico genómico aquí en INMEGEN. Entonces, ellos van a estar aquí de 11 a 12. Va a llegar más gente a este auditorio y bueno, en orden, de modo que podamos acomodar esta presentación, vamos a hacer un break ahora de 30 minutos. Tenemos servicio de café que solamente va a estar disponible por 15 minutos porque tenemos que remover por la gente adicional que llega. Entonces les invito a tomar un café. Tenemos café, galletas y nos vemos de vuelta aquí a las 11 y